Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome back to the deep dive. Okay, let's get right into it. If you've ever looked at a genetic test result, you know the labels. Pathogenic, benign, or that really tough one, VUS variant of uncertain significance.
Right. And the big takeaway from this major study we're digging into today is that those labels, they're just incomplete. The really critical information isn't the category, it's a number.
A specific number, yeah.
Yeah.
A probability of risk.
Exactly. A precise risk percentage. This study used machine learning and data from over 1 million electronic health records to basically translate those vague labels into actual numbers. They call it ML Penetrance, or MLP.
So we're shifting from just sorting variants into boxes towards like calculating a specific risk score for individuals for dozens of disease genes, too.
It really changes the game. I mean, think about it. Figuring out the risk for a rare variant used to take ages, right? Small studies, families, very specific situation.
Limited scope.
Totally.
Yeah.
This gives us a way to do it at scale, quantitatively, overcoming a lot of those old hurdles.
Okay, so let's talk about those hurdles first. Why were we stuck with these labels and, why aren't they good enough, especially when the stakes are high?
Well, a big part of it is that VUS category variance of uncertain significance.
Yeah.
It just causes so much clinical anxiety.
For doctors and patients, I bet.
Absolutely. You have experts disagreeing on classification, patients left wondering. It's a really difficult gray area. No one can give them a solid number.
And even for the variants labeled pathogenic, the ones we think we understand, the risk isn't always clear-cut.
That's the other key issue, ascertainment bias. It's a bit of a mouthful, but basically, traditional studies often focused on families where a disease was already really common or really severe.
So they were looking at the worst-case scenarios, essentially.
Precisely. And if you calculate the risk based only on those families, you're going to overestimate how likely someone in the general population is to actually get the disease. A variant might seem 90% penetrant in that specific family context.
But maybe it's only, 20% in reality?
It could be, yeah. Much lower when you look broadly, so you end up inflating the risk estimates.
Okay, so we need two things then. We need scale, lots of people, not just specific disease groups.
Yeah.
And we need deeper health information, right? Not just yes, no on a diagnosis.
That's where the electronic health records come in. The EHRs. Over 1 million of them in this study.
That's massive scale.
It is. And it gives you what's called deep phenotyping. You're not just looking at one snapshot, one diagnosis code. You're seeing years, maybe decades, of routine medical data. Lab tests, vital signs.
The whole picture.
Right. It lets you model disease as more of a spectrum, which is much closer to reality than just sick or not sick.
Okay, but quick question. If you're using EHR data, aren't you still looking at people who, actually go to the doctor? Isn't there still some bias there? People who seek healthcare might be different.
That's a really important point, and yeah, you can't eliminate all selection bias, but it's mitigated here in a couple of ways. First, the sheer number, over 1.3 million people across different health centers, helps average things out. And second, crucially, it's what data they train the machine learning models on. That makes a huge difference.
Right, let's get into that. The ML blueprint, because you said they didn't just try to predict diagnosis codes?
No, definitely not. That would sort of bake in the old problems. Instead, they built these pretty sophisticated ML models using something called extreme gradient boosted trees to generate a continuous score. A disease probability score from 0 to 1.
Based on what, though?
Based purely on the kind of objective, routine clinical data you get all the time. Things like LDL cholesterol levels, kidney function tests like GFR, hemoglobin counts, vital signs.
So the standard stuff from a physical, not doctor's notes or billing codes for diagnoses.
Exactly. They specifically avoided using diagnosis codes as the main features for training the core models. This forced the ML to get really good at spotting the underlying biological state associated with a disease.
So it learns to recognize, say, the pattern of labs that signals early polycystic kidney disease, even before a formal diagnosis is maybe even made.
You got it. It's looking at the biological signals, and these models were trained and validated very carefully across huge, separate groups of patients within that 1.3 million total.
Okay, so you have these validated models that can score someone's disease probability based on their labs and vitals. How do you get the MLP, the risk number for a specific variant?
So they took these validated models and applied them to a completely independent group of people, folks who had their exomes sequenced, so their genetic data was known.
Right.
They looked at about 1,648 rare variants across 31 different disease genes. And for each person carrying one of these variants, they generated that continuous disease score using the ML model. The MLP is essentially derived from comparing the scores of people with the variant to those without it.
The People's Health Series, right? How do you get one single number?
Good question. They used some clever math, basically accounting for individual variations, how the model performed on different runs to refine it all down. It filters out the noise to give one stable, reliable risk probability for that specific variant.
Wow, okay. So it's turning all that messy, real-world clinical history into one actionable number.
That's the goal. A clean signal from noisy data.
Which brings us to the big question. Did it work? Did this MLP number actually match up with what we see in patients? Validation time.
Yeah, and the initial checks were really encouraging. The variants that we already knew were pathogenic. The bad ones had a significantly higher median MLP, around 0.52.
Okay, that makes sense. And the benign ones?
Much lower, as you'd expect. Median MLP was about 0.28, so the basic separation worked. Rare variants also generally had higher MLP scores than more common ones.
But you said the real insight wasn't just the separation, it was looking inside those categories, like within the pathogenic group.
This is absolutely critical. Even among variants already labeled pathogenic, the MLP scores weren't all clustered together. There was huge variability. Some pathogenic variants had much lower MLP scores than others.
So just calling something pathogenic isn't enough. It doesn't tell you how pathogenic, like what the actual risk percentage is.
Exactly. That label hides a whole spectrum of risk. Is it a 40% chance of disease or an 80% chance? That difference matters hugely for clinical decisions. MLP gives you that number.
So not all pathogenic variants are equally risky in the real world. Did the MLP score correlate with actual bad outcomes for patients, like serious health problems?
Yes, they checked that carefully. They looked at clear measures of disease severity. For instance, polycystic kidney disease, or PKD. For people carrying PKD variants, every 0.1 increase in their variant's MLP score meant about a 9% higher odds of developing end-stage renal disease.
Wow, needing dialysis or a transplant. That's a direct link between the ML number and a severe outcome.
It is. And they saw similar things for familial hypercholesterolemia, the high cholesterol condition. A 0.1 increase in MLP there correlated with about a 2% higher odds of having a heart attack. Higher calculated risk, worse outcomes down the line.
Okay, that connects it to patient health. Do they also link it back to the underlying biology, like what's happening in the cells?
They did, which is really important for confirming it's not just some statistical fluke. They looked at BRCA1 variants. Those are important for DNA repair, related to cancer risk.
Right.
They found that higher MLP scores for BRCA1 variants significantly correlated with lower scores on lab tests measuring actual DNA repair function. So the clinical risk score derived from EHR data reflects a real breakdown at the molecular level.
That's pretty compelling. It connects the dots from the clinic back to the lab bench. Okay, let's talk utility. How does this help with that VUS problem, the variance of uncertain significance? That's where so much anxiety lies.
Yeah, MLP seems really promising here. When they looked at the MLP scores for VUS variants, the median value fell right in the middle, around 0.46, sitting between the typical benign and pathogenic scores.
So it immediately gives clinicians a kind of probability estimate, instead of just uncertain.
Exactly. It provides a starting point, a quantitative measure to help triage these ambiguous variants. That alone could be a huge relief.
And how did MLP compare to the older way of estimating risk, that diagnosis-based penetrance, or DX penetrance?
Oh, the comparison was stark. DX penetrance, because it just relies on counting diagnosed cases, gives really coarse, blocky results, especially for rare variants. If you only have, say, 3 carriers in your study.
Your only possible penetrance values are 0%, 33%, 67%, or 100%.
Right. It's these big, unhelpful jumps, not very useful clinically. MLP gives you that smooth, continuous distribution of risk scores instead.
A much finer picture.
Much finer. And importantly, the MLP method was able to provide risk estimates for about 20% more carriers who just got excluded in entirely by the rigid rules needed for the old DX penetrance calculation.
Okay, let's make this concrete. The paper showed specific examples for VUS and polycystic kidney disease, right?
Right.
Using that long-term clinical data you mentioned.
Yes, this is where it gets really powerful.
Ah.
They tracked carriers of PKD VUS over time. They found someone with a VUS that had a high MLP score of 0.89, so almost pathogenic level risk. And that person showed, over years, a clear, steady decline in their kidney function tests, their GFR, their hemoglobin levels, textbook signs of progressing kidney disease. And indeed, they ended up with multiple kidney disease diagnoses.
So the high MLP score predicted that outcome, that VUS was effectively pathogenic.
Correct. Now, contrast that with another person who had a PKD VUS with a low MLP score down at 0.17, so closer to benign risk.
And what happened to them over time?
Nothing significant. Their kidney function markers, their hemoglobin, all remained stable over the same period. They never developed a kidney disease diagnosis.
Wow. So the MLP score, derived from population data, accurately predicted the individual long-term clinical trajectory for both of these uncertain variants.
That's the key insight. By integrating that longitudinal data watching the movie, not just the snapshot, MLP can help separate the truly damaging VUS from the harmless ones. That resolves a huge amount of uncertainty.
This feels like a really practical tool.
Well, the framework itself, this MLP approach, it's designed to be a scalable blueprint. It leans heavily on data that's already being collected, routinely lab tests, vitals, relatively cheap, readily available data.
Right. Not requiring specialized, expensive tests for every variant interpretation.
Exactly. It captures that disease spectrum across different levels, from biomarkers right up to organ function, in a way that simple case control studies just can't.
And the implications for actually using this in medicine, for precision medicine.
I think they're pretty immediate. Imagine a patient comes in with a VUS in, say, the BRCA2 gene. High stakes, right? Cancer risk.
Very high stakes.
Instead of just saying, it's uncertain, we'll watch you, you calculate their specific MLP risk for that variant. Maybe you set a threshold, say if the MLP is over 50%, then that person gets flagged for enhanced screening. Earlier mammograms, maybe more frequent MRIs.
But if their MLP is low?
If it's low, maybe they can stick to standard screening protocols, avoiding unnecessary procedures and the anxiety that comes with them. It allows for truly personalized risk stratification based on a concrete number.
That makes a lot of sense. Where does this go next? What's the future for this kind of approach?
Well, there are definitely crucial next steps. A big one is expanding this to diverse populations. This initial study was largely based on European ancestry individuals.
Right. We know genetic risk can vary across different ancestries.
Hugely. So developing and validating ancestry-specific models is absolutely essential for health equity. Otherwise, the tool only helps some people. That has to be a priority.
Well,
Also incorporating other factors. Right now, it's mainly focused on the single variance effect, but we know genes don't act alone. Future models could try to bring in things like other genetic modifiers, environmental exposures, lifestyle factors.
building an even more complete risk picture.
Exactly. Getting closer to that truly comprehensive view. And of course, extending the approach beyond the initial set of diseases studied here.
So wrapping this up, what's the fundamental shift here for someone listening? What's the big takeaway?
I think the bottom line is this. ML penetrance moves genetic interpretation away from just asking, is this variant bad or not? That static yes-no question.
Towards something more practical.
Towards the truly practical question. For this specific person carrying this specific variant, what is their actual quantifiable risk of developing the disease over time? It's about personalized, numerical risk.
And that leads nicely into our final thought for you, the listener.
Yeah, think about this. MLP worked by integrating all that continuous health data, your lab results, your vital signs over years, into a single risk score. What if, eventually, machine learning could define disease itself, not just by a sudden diagnosis code appearing in your chart, but by detecting those tiny continuous shifts in your own unique health metrics tracked over your entire lifespan? Maybe the diagnosis becomes less of a single event and more of a gradual, personalized warning flag based on your trajectory, detected far earlier.
Defining disease by the pattern over time, not just the endpoint. That's definitely something to think about.